that where some of the undergrads and other developers have worked with Cytoscape and GenMap and Wiki Pathways. Then he launched Wiki Pathways in 2007 with a small research program at Gladstone Institute in San Francisco, and is currently an associate director of bioinformatics at the Institute and executive director of the National Resource for Network Biology. So with this further ado, I'll just pass it on to Alex for his presentation. Looking forward to it.
Great, thanks Pankaj. Thanks Lisa. Thanks for inviting me to speak and for the introduction. What I'm going to do today, I usually, when I talk about Wiki Pathways, it's to a group of researchers who are primarily interested in using the content for their research and maybe secondarily interested in participating in BioCuration, and most often not interested at all in sort of what's under the hood and how it works and how we built it. And so I gathered from the invitation and the setup here, you guys actually do want to hear a bit more behind the scenes. And so it's going to be a mix. I'm going to first introduce Wiki Pathways, what it is and where we're at today. And then I pulled up some slides from about 6 years ago that gave some early perspective on how our first 4 years went, basically. And so with that context, I'll share some of the lessons we learned as we got this thing initially launched. So hopefully aspects of that are relevant to you know, cautionary tales and encouragement for trying to do the same for new wiki or community-based efforts. And then I'll touch on there's lots of different directions our conversation could go today. I'll touch on some of the developer aspects, some of the BioCurator aspects, and some of the end user aspects. I won't go into too much detail and leave some time for questions and discussions if you want to dive deeper into any of those particular branches. I'll be happy to take those. All right. And feel free to unmute and interrupt at any point if something's not clear, if you have a question. Okay, so what is Wiki Pathways? It's a pathway database like KEGG or Reactome, if you're familiar with those. And like any pathway database, one of the most important numbers is that you have a lot of pathways, and we do. We have all the same canonical pathways that you'd find at Reactome or KEGG. But what's unique about our pathway database is that we have a lot more activity and a lot more editors than a typical pathway database. You probably know this already, that most biology databases are managed by a small group that's internally funded to sort of curate and keep things up to date. You might be pulling information from lots of places, but the actual editors or authors are a select few. And so we took the wiki model. We literally took the MediaWiki platform that Wikipedia uses, and we ripped out the text editor that people use to write encyclopedia articles, and we put in a pathway drawing tool, so a simple diagramming tool. And so now you're drawing and editing pathway diagrams, clicking save, and they're then shareable with others who can then edit them and add to them over time. And with this approach, we're finding that we're able to cover more pathway types and more gene coverage at this point than any other database. And so we're able to keep up with the influx of new pathway information much better with this approach than we were when we were a traditional database prior to Wiki Pathways. So each year we're seeing something like hundreds of new registered authors each year. You only have to register if you want to edit. So this doesn't count folks who just want to download things or browse. Each year we get about 3,500 edits to over 1,000 pathways. To contrast this with KEGG, which has been the gold standard pathway database, you know, they're making dozens of edits a year by maybe a half dozen people, and so it's just hard with that sort of with that approach to keep up, and so that's one advantage of this approach. each month, on the other end of things, in terms of consumption and visitors, lots of people visit the website, but even more folks use our content programmatically. And so we have a web service and API, so folks can download all the pathways at any time and perform bulk analyses. And that's the most common use of pathway databases in general. The content is also distributed into databases and analysis tools that are already freestanding and hosted by other sites, and we're not really tracking that usage at all. So those are a bunch of numbers. If you're more of a visual person, you know, I think this plot is pretty clear. We stopped updating it at some point because we sort of passed this goal and are now trying to measure ourselves by different metrics. But you can see here that KEG, after they switched their licensing model, after they lost their national funding, has had a tough time continuing to grow and stay up to date, whereas our model had a much later start, but it's on a completely different trajectory. So it's very much not like traditional databases when you think about it in these sorts of contexts. Okay, so that's hopefully enough introduction to sort of what Wiki Pathways is today. It's been up for just over 10 years, and that's sort of where we're at and today. And so now I want to look under the hood, like I mentioned before. And there's a few aspects I want to go into detail in. The first is the BioCuration of pathways. I think there's some content here that's relevant to your group and what you might what endeavors you might be taking on in terms of attracting a community of contributors and how to do quality control, etc. And some lessons learned about working with existing standards and connecting to existing workflows. I'll reiterate some of those. And then some of the aspirations of an effort. You know, when you want to make a non-traditional type of database, there's challenges and there's some potential to make positive changes to the culture of research. And we're still working on that as well. Okay, so BioCuration, the overall picture is represented here. On the left are sort of the inputs and on the right are the outputs. And so a biocurator will read papers. Some of our biocurators are incorporating experimental data from their own labs. And then static figures, there's about 500 pathway figures published each month. And those are just published and drawn in PowerPoint or Illustrator. And so they're completely inaccessible to search indexes or to programmatic analysis tools, and basically hidden away, and only the individuals who happen to read the paper will ever see that information again. And so we think that's a real shame. And so one of our ongoing efforts is to pull that data back out of the papers and model it properly in Wiki Pathways so that you get these outputs, right? You get diagrams that are human readable and citable. And so there's authorship associated just with the diagram itself. But of course, the diagram can be used for data visualization to be produced as an image, a publication quality figure image. And at the same time you're drawing the image, you're actually building an XML model. I'll talk about that more later. And so that you're creating a computer readable document at the same time that's then amenable to visualization and analysis tools, tools like Cytoscape, which was mentioned in the introduction. It's a collaborative tool, and we freely distribute the content with a Creative Commons 0 attribution to as many tools that can use pathway information. And so a typical, just looking at an example pathway, this is an EMT-specific colorectal cancer pathway. At a high level, without looking at the details yet, you can sort of see the structure of a typical pathway page at Wiki Pathways. So there's the interactive diagram at the top. This one includes almost 200 genes and a bunch of interactions. So it's rich content in a particular pathway. We then have a free text description section where authors can describe the pathway in a natural language that they would use to describe it to colleagues. But together with that, we have some structured information. We have what we call quality tags or quality assurance tags. There's probably 24 different types of tags, but the most important one is this approved version tag. And that allows us to, if there's a new edit made, before it gets distributed to all the tools, it needs to be approved by a second author before it gets integrated into other analysis pipelines. And so that helps catch mistakes or anything that might be disputed. The other structured information are the ontology terms. So in addition to the description, here you can specify specific terms from 3 different ontologies. We have a pathway ontology, a disease ontology, and cell type ontology. We pulled these 3 from the hundreds that are available at BioPortal. I think they've renamed it. It's that Stanford group, used to be called BioPortal. And I would recommend them as a resource for if you want to annotate any of the databases you're thinking of spinning up with ontologies. That relationship's worked out really well. And then each paper, right, is composed of from studying a number of publications, each pathway. And so this one has 8 publications currently associated with it. So there's a bibliography that naturally is constructed as you build the pathway. Now, if we zoom in a bit, on the left here, I'm showing a little inset area that we're then zooming into on the right, just to show you one corner of the pathway to see a bit more detail. The boxes are, we call them data nodes. They can represent genes, proteins. They can specifically, like in the bottom right, represent microRNAs, microRNA species, or other pathways. And so each of these boxes is actually connected to a primary database, a database like Ensemble or Entree or UniProt, for example. There's dozens of databases you can choose from. And that's part of the process of making sure that these are computer readable and not just human readable. So the human will read twist 1 and twist 2, for example, but connected to that in the model is a database identifier, for example, Ensemble identifier, and computationally you can search for that, overlay data on that, and perform pathway enrichment analysis. We also have interactions, of course. You'll see a number of different interaction types in this inset. Just a standard arrowhead is usually like an activation. There's the open circle for catalysis, the red T-bar for inhibition. So those are very natural semantics. There's also, for SNAI2, 1 and 2, there's a transcriptional relationship. And so there's a lot of semantics that are encoded in the interactions. Then you also notice the phosphorylation states, these circled P's on some of these proteins. Again, those aren't just graphical, human readable only. Each of those circled P's has data associated with it, like exactly which residue at which position. So it can refer to tyrosine 254 on this particular protein, and whether the phosphorylation at that position has an up or down regulatory effect on the normal function of the protein. All of that is captured in the computer model as you draw the diagram, again, so that it can be used in analysis. All right, that's what a pathway model looks like. I've mentioned this a few times, but just to sort of drive it home, because it's a subtle thing that happens, and I think it's really important distinction between modeling a pathway and just drawing it in Illustrator. In the middle is what is sort of a cramped view of what our drawing tool looks like. You can interactively add from the palette, add different objects and connect them, all using mouse controls that are very natural, like any drawing tool. But in the right is an information panel, and as you're connecting these nodes, these data nodes to information, you're actually, on the far right, you're actually generating an XML document, an XML model that you never have to look at, but it's important to know that you're getting both the image on the left and the model on the right as you're using our drawing tool. They're built simultaneously. And whenever you make an edit, you have to use this same drawing tool so that you're updating both the XML model and the image at the same time. And so what that gets you is it's an open standard, our XML format. It's called GPML, and so it can be converted to BioPax or any other SBML or any other pathway format that you might be interested in. We also have a REST web service API to be able to access these data. The models can be embedded into web pages with a simple iframe, a one-liner in HTML. We provide the technology to give you an interactive pathway view on any third-party website. And then we also have an RDF representation, so a more formally semantic representation of the content, and a Sparkle endpoint for querying that. And so that's a lot of the sort of what a lot of the back-end data modeling gets you. That's beyond just a static image. Okay, as a specific example of something you do with the data model is to pull it into Cytoscape. It's a little cut off. Sorry about that. This is one of the older slides I tried to pull in. In the upper left, you can actually see a pathway diagram in Cytoscape that's imported as a pathway, meaning that it maintains the layout and look so that it looks identical to the human curated layout that the original has at Wiki Pathways. But in the center, what was supposed to be the lower right, you can also import the pathway information as an abstracted network. Cytoscape more typically works with abstracted networks, and that lets you apply algorithmic layouts. It allows you to extend or try to find shortest paths, identify hubs. You can apply 100 years of graph theory through network analytics to these pathway models in Cytoscape when you import them as networks. So it's again a nice flexibility that having a proper data model gives you, is you can have alternate representations of the same information. I mentioned the RDF representations, and here is abstracted network representations, which are very useful for analysis. Okay, so there's sort of, this was one of the first slides we made for Wiki Pathways. I'm pulling out some old slides now. And there's this virtuous cycle that we're going for here, right? Where we're modeling data from the published literature, modeling it in these pathway diagrams that then allow for synthesis with other data, for data overlays, or with other interaction data. You can use tools like Cytoscape to do that. Visualizing your data in the context of pathway models is then a great way to formulate new ideas and come up with new hypotheses. And then it goes back into the lab where the researchers then can go and generate more data. And I tried to pick, I did a generic search for scientist in Google Images back when I made this slide, and this was the #1 image. So it was an unbiased selection. We have a long way to go still with our reputation, I guess. And so this cycle's working, and we can see that in the growth curve of Wiki Pathways. So before I talk about that, the other aspect that you might want to discuss more, and I'm open to it, I tried to capture it in this slide, has to do with quality assurance, quality control, and moderation when you're doing a community effort. Pankaj knows about this as well, and maybe you guys have experience with trying to corral and recruit community contributions. And so from the Wiki Pathways perspective, there were a few lessons we learned and approaches we took that I'll talk about here. The first are just from the start, we were following Wikipedia's lead. When we started the project in 2007, it was right around the time when that paper came out in Nature that compared Wikipedia against a standard encyclopedia in terms of quality of information. And a lot of people were surprised, and a lot of people disbelieved, and some people still disbelieve that you can get anything of quality out of Wikipedia. Some people have the inherent bias that, oh, well, if anyone can edit it, then you're going to end up with garbage. These sorts of things should only be done by quote unquote experts. And so we disagreed with that. We had been maintaining a database as experts for a number of years at that point, and we're really frustrated about how slow it was and how limited we felt. You can't hire a team big enough to cover all of biology, to be an expert in all of biology. It's just not possible. And so we were inspired by Wikipedia's model. And so, like I mentioned, we simply took their open source software and swapped in our pathway drawing tool, and tried to not change as much as possible. And so we didn't recreate our own curation model. We actually used the curation models that they have for tagging things, for creating watch lists, for defining roles, for administrators, they call them bureaucrats, versus contributors, so that there's a sort of an organization to the community that's just inherent in using this tool. And so we reused a lot of that from the Wikipedia model, and it's worked out really well. One contrast, for example, an advantage we have, and I think you'll have as well, as a biology niche database or community content coordinator, is that you don't have 13-year-old boys who want to mess up your pages like they do at Wikipedia. And so in the 10 years that we've been running, we haven't had a single act of vandalism, for example. People have made mistakes, and we're able to use the same mechanisms that Wikipedia established to quickly find and fix the mistakes before they propagate. And all of that's working really well. They have a whole dispute mechanism, and we haven't even had to use that yet. And so a lot of the pitfalls and challenges that Wikipedia's had, we're prepared for them, and we haven't even had them. So in that regard, the model has worked really well. I mentioned watch list, so that's sort of the first order of defense or of quality assurance. Anyone who edits a pathway or creates a pathway is by default added. The pathway's added to their watch list, which means that if anyone makes subsequent edits, they're notified by email. They can remove that, obviously, if they don't want to be notified. But that's a nice way to sort of assume a level of interest in the content, and you can then check what was edited and dispute it or undo it if you think it's wrong, or contribute to approving it if you think it's right. You can also voluntarily add pathways to your watch list as well. You don't have to edit them. And so that's part of the community quality assurance. We also assign a curator of the week. This is something we put together ourselves just because we realized we were sort of doing it naturally. And so we just made it a formal thing so that there weren't weeks where we just happened to miss. And this individual is responsible for tracking every edit that's made to any pathway in a given week. And so they usually do split that over 2 different days during their assigned week. And that's worked really well to just, again, not catch any vandalism, but to just catch mistakes, or to find a situation where there's a new author who looks like they're going down the wrong direction and they don't understand the tool, or they're frustrated, and you can kind of catch them early and say, hey, here's some help documentation, or how can we help you? And so it's also sort of a help desk as well. And then, the most important thing is that you identify before you start a project like this that you even have a community of stakeholders. I'll reiterate this a few times, but it's not the case that if you build it, they will come. That does not apply to biological databases or community efforts in biology, in our experience. Our project has worked well. One of the main reason why is because the community was already there. The needs were already there, the workflows, the idea of pathway models, the use cases for pathway models. were all in place. And so we were just providing a tool and a new mechanism and perspective for existing communities, and then formalizing some of the organization and connections in that community. But we weren't trying to create a community from scratch. I've seen a lot of biology-based wikis come and go over the years, and that's one of the major determining factors, is whether they're trying to do something from scratch or play into communities that already existed. Okay, so going way back into our history, these were these are about 6-year-old slides, just to show you what our perspective was like about starting Wiki Pathways. If you're at that stage of deciding whether to start, or at the early stage of a community project, this is what it looked like for us. And so, like I had mentioned, we had been maintaining pathway databases for many years prior to Wiki Pathways. As far back as the project started in 1998, actually. And the growth curve, I didn't bother plotting all of it. The growth curve looked pretty much like this. It had an initial bump, obviously, to go from 0 to about 100 or so, 120 or so pathways, which were the major canonical pathways at the time. And then it just plateaued, and it wasn't really growing. We noticed that people were using the tools to draw new pathways and even publish them, but they weren't sending them back to our database so that we could grow the content and distribute it centrally. And so we were using wikis at the time for a lot of project organization. We had the idea in 2007, let's make a wiki for our pathway database. And so that was the idea. Around 2008 is the official start of Wiki Pathways, and you can see it just completely changed, a completely different paradigm for growth and content maintenance. And it's a little harder to measure, but no one on the team disputes it. None of our users dispute it, that the quality is actually increased as well. So it's not like we're trading off quantity for quality. We're actually getting higher quality. It's easier to make corrections. There's more eyes on every pathway, and we're getting more pathways and more content. And so this was just the first 4 years. The growth curves continued to go up like this, as you saw in the contrast with KEGG. Another important measure, so this is just the period of Wiki Pathways from 2007 to 2008. I'm not showing, where it's just sort of mostly an internal tool still. And then in 2008 is when it really became a public tool. And this is what our initial counts looked like for people who are registering as users and people who are actually making edits. And this percentage was pretty good, actually. It was more than we were expecting. It's much higher than what Wikipedia has. But that's not too surprising, because we are working in a niche. Most of our users are qualified to be curators, whereas most of the users of a Wikipedia article probably aren't qualified to edit and don't have the knowledge to edit every article they read. At some point, though, we stopped tracking this number, because you can see as this the registered users did receive some bloat at the end, and we later realized there's a bunch of bot accounts, and so we had to figure out how to filter those out, and so this tracking this further became problematic. But the editing users is a real number, and that's just steadily inclined. That hasn't changed its shape at all. We would have liked to see it inflect up, but we haven't figured out how to do that. And so another challenge with the project that like this that we faced and others have faced, and you'll likely face, is just sort of make sure you're setting your expectations right for how many external contributors you'll actually get. It likely won't meet your most your highest ideal numbers. Okay, a couple more. A couple more lessons learned in the early days. The most important one for us that got us through that first year and a half was to set an early milestone. And so we went online in March of 2007. but it was mostly an internal tool until January of 08, right? So that's 9 months before we had someone log into the system that none of us knew, right? And so that's quite a long time. If this was the primary, if the primary only use of the tool was to be a community-driven tool, it might have died during that time, that 9 months. But the early milestones that we set was actually that the tool was useful for us internally. We were already curating pathway content for many years. And even if no one had ever logged on to Wiki Pathways, we would have kept it as our own internal editing tool because it made our lives easier. And so I think that's an important lesson, is that whatever tool you're going to build, make sure you're designing it in a way and fulfilling existing needs and communities in a way that it's going to be useful right out of the bat. Even if no one else signs up for it, there would be a reason to keep hosting it and keep maintaining it because there's an immediate demand and immediate need from at least some group of people and who are already doing that type of work and not just promising to do it, because it's going to take a while to build a community. So yeah, that's what we call the birthday of Wiki Pathways, is when we had our first unknown user, which again was about 9 months after we conceived of it. Okay, and then some other things that I would say not to try to be too ambitious with. If you're going to do a community-driven project, it's important to work with established models or standards, like we did with biopacks and our XML file formats. I mentioned this already. Working with existing communities is really important. Instead of thinking about building ones from scratch, you got to identify existing ones and see what tools they need today. Working with existing tools and pipelines. So pathway analysis is our niche. We're making the pathway database. We have to fit our content into existing tools and existing workflows, bioinformatics workflows. We can't just expect everyone to swap out all their tools and swap out all their scripts to use some new sort of thing that we're just releasing. We'll get people do this all the time with grad student projects, and it gets them that paper and lets them move on with their project. But then once that developer's gone, the tool will disappear. And so doing the research to find existing tools is important. Publishing models has been a tricky one. We're still struggling with that. So by far the majority of pathway figures that you see published today are still drawn in Illustrator or PowerPoint. It's frustrating. We're providing an alternative. It's almost as easy to draw in Wiki Pathways as it is in PowerPoint or Illustrator. You have to do a bit more work because we want you to actually model it, right? Not only draw it. But we think the benefits outweigh that burden, but now we're getting into cultural shifts, which are tough and move slower than you'd like. And so we've talked to different publishing houses, and they all like the idea of incorporating pathway modeling into their publication process, or at least even recommending it, but there's been zero movement on actually establishing these sorts of things. And so, we're still fighting that one. And so I think the lesson here is that if your product depends on changing a major cultural institution right away, it's going to be tough. And I would discourage investing a lot in that, a lot of time in that, because it's not likely to work. But having said that, I think it's important to have those aspirations. So while your project shouldn't depend on them, I think it's important to push on those boundaries. So, tweaking the established models. We didn't just adopt an existing pathway model. We did design our own open standard. We just made sure that it was compatible with existing models, but we did tweak it. we're trying to grow the community. We're trying to convince more people that they're capable of being biocurators. A lot of the data consumers have the knowledge in their head to fix and grow and contribute to pathways, and a lot of them aren't. And so we're still pushing on that boundary. And changing perspectives about publishing is an ongoing effort. One of those perspectives is the one I just mentioned. Everyone is a curator, potential curator, and that the knowledge should be open. This is a lesson that we're learning with KEG. Once they lost their funding model, they then were able to switch their licensing, and now technically you're supposed to pay if you want to do bulk analysis with KEG. And our content is open source and open access and licensed as CC0, which is even better than public domain. And so there's no way we could ever charge or lock down the content. It's guaranteed to be free and open. I think that's important for community projects where you're asking people to volunteer their time and knowledge into a database. I think an important part of that that exchange is to guarantee to them that they and their colleagues will always have free access to the knowledge that they've contributed. You're not paying them; they shouldn't have to pay you later. Okay, so those are the slides I had prepared. This is our old acknowledgement slide, again from 6 years old, which is sort of a nice one. So it lists here all the people who were involved in the original creation of Wiki Pathways. The PIs at the time were Chris Evelo in the Netherlands and Bruce Conklin here at the Gladstone. And then we had just started the National Resource for Network Biology and R&B and participating in Google Summer of Code. We started Google Summer of Code, our first year was 2007, I think. This is now our 11th year doing it. Yeah, I think that adds up, right? And that's a program that Pankaj mentioned where Google pays students around the world to write open source code for approved open source organizations. And through Wiki Pathways and NRNB, we've been able to participate as a mentoring organization for 11 years. Each year we get between 10 and 20 students writing code on our projects, for our projects paid for by Google. And so we're doing that again this summer. And then I have an R01 grant from NIGMS purely for Wiki Pathways development. I just had that renewed last year. And so we've got nice stable NIH funding for the project for the next while. But we are brainstorming on what the long-term funding model is and still haven't figured that out. It can't be funded by R01s forever. And so we can talk about that if you want. And then on the Netherlands side, this has been a collaboration from the start with Chris Evelo's group, and he runs a group called Big Cat Bioinformatics at the University of Maastricht in the Netherlands. And then this is something I set up 6 years ago, so it's not up to date, but it's still kind of fun to play. While I take questions, I usually run this running scroll of contributors. So these are all the authors that had contributed to any of the pathways at the time. So I don't know, there's probably 250 names or something like that on this list. So I'll stop there and feel free to unmute, and happy to take questions for a while.
Wow, that was quite something, Alex. Wow, it's very impressive. I love the scrolling names.
So this is Jackie here. I have a question. How do you mentioned that you contact or collaborate with your community. How do you do that? Do you do it via email or is it through conferences? Because it's one of those things where you can send an email, but the likelihood of someone even opening it is maybe 50-50.
For the email notification for watch list changes?
Yeah.
Yeah. Yeah. So I mean, the point there is to have a system in place that if someone cares, they manage their watch list, right? Mm-hmm. So if they have an interest in this pathway or that pathway, so say they drew the pathway or made a major change to it, and maybe they've even published it as a figure in their paper. This is the model, for a lot of labs today. A pathway model is sort of is the dogma that their lab is working on, and it's the basis for their grants and for all their research. And so they care a lot about that pathway model. And so it's a mechanism for them to get email notifications when it changes. So the point isn't to sort of spam a large group and kind of cold call them and hope they happen to open the email and care about it. It's allowing them to get notifications when it changes, so that they don't have to go and look at the page all the time. And so, yeah, if someone doesn't want notifications, they can turn them off or not open them. That's fine. But the point is to allow folks who do care about the content changes to look at them. And that's, again, just one of the layers of QC on edits.
Hi, this is Lenore. Hi. Can you, so about how much time do the curators end up spending on the actual curation? Like, I imagine there's sort of different levels, right? There might be somebody who's come in and it's like, I'm going to put in a whole new pathway that doesn't exist. I suppose that's rare versus the edits. Mm-hmm. So, I mean, in our experience, right, authors will contribute, but they do have difficulties. So there's always some finessing. Mm-hmm. And I'm just wondering how involved the curation is for Wiki Pathways.
Yeah. So to start a pathway from scratch, for someone who's done it a few times, I'd say it takes about 4 hours from scratch to draw a complete pathway and model it with all the database connections. It takes me or the other curators that have been doing this for a while, probably about 4 hours. Often what you see from folks who this isn't their job, right? So there's some bench biologist and they're gonna build a model. They usually do it over a number of days. And so they'll spend an hour on it to kind of get, the first core of it up, and then they'll come back to it a couple days later and add a bit more. And so there's an under construction tag is one of the quality assurance tags. And by default, that's just added to all new pathways. And so while it's under construction, it's visible to other people if you search for it, but it's not automatically sent out, to be distributed as part of the collection. And so people can build a model over, anywhere from 4 hours to maybe 2 weeks, depending on how frequent you want to work on it and how much experience you have. And then you're right, there's also that's building one from scratch. There are lots of folks who just find a mistake and want to fix it, or some people, their entire PhD, the result of it boils down to adding one more data node to an existing pathway and drawing a connection, right? And so, that takes minutes to do.
Do they get, do you assign DOIs to the pathways? Is there?
Yeah, the pathways are citable by URL right now. You can, there's some steps you can take to produce a DOI if that's what you want, but we don't automatically do it yet. We've talked about it, but there hasn't been much demand for it. Usually folks just want a URL to put in their paper. The URL goes to a specific version of the pathway, so that if it's changed in the future and updated, your paper can always reference, if you want, the version and exactly how it was at the time you produced that URL. So I think it's better than the DOI, frankly.
I can vouch for some of the things that I've done, and what Alex just mentioned. I can take anywhere from 15-20 minutes. You read a paper and there's a new gene interaction showing up. You go and add it, and it's more or less like as if you're drawing a pathway in a PowerPoint. It's more or less like that, but a little bit more of data tracking behind the scene that happens. So and any user can learn it. I think half an hour, 45 minutes is more than enough to get going, and then and start drawing on that side kind of things. I don't think he's shown something, but there is a gene expression analysis that can be done. I believe it's there, and then I don't know if it still exists or not. I haven't used it for a long time. But there was a probability of using all the pathways from that species and carry out your gene expression analysis, right, Alex?
Yeah, so that's one where that's not built into Wiki Pathways, but there's existing tools out there. So Enricher, you can use Cytoscape, PathVisio is maybe the one you're thinking of. That's one we do.
Yes.
Yeah, and so there's a lot of tools that do enrichment analysis on pathways, and all of those tools I mentioned and many more, we have a page listing them. They pull in Wiki Pathways on a regular basis and just provide it as one of the pathway databases together with gene ontology and other ontologies to do enrichment analysis again. That's the most common use of our content, probably. Mm-hmm. And on the editing topic, I'll put in the link in a chat here to something we just built a couple of years ago called Academy, Wiki Pathways Academy. That's how, what we wanted to do with that is, the idea is starting from scratch. I think anyone, any high school student or anyone with basic biology knowledge should be able to start at the beginning of this path. If you, like many of us here, already have advanced biology knowledge, you can probably skip the Bio 101 track, for example, and just skip the path down to learning how to use the tool. And it starts from the very basics of just adding one gene to a pathway and connecting it to a database, and then adding a second gene and drawing a connection between them. And it just takes you step by step. Each lesson or each step in the path just takes a few minutes, and you can break it up. So you can do a path and then take a break and come back a couple days later and do the next path. And by the end of this training series, you can build a whole pathway from scratch, starting with
I'm sorry.
Whoa.
So I shared that link. So, if you want to get a sense of what it takes to edit the pathways, I think that's a good one training mechanism to share.
Alex, this is Rex Nelson. I work at Soybase. And I'm how do you get started from scratch? I see you've got glycine max as a browsable species, but it seems to be empty. Do you stub it with something, or do you just start at scratch?
Yeah, good question. So in terms of new species, so when you look at it in the browse, by default the tags are showing you probably featured or maybe approved. And so if it's showing you only the featured ones, maybe that's why it's empty. None of the species should actually be completely empty, because what we do is we add a new species when we find a community, an existing community that's studying that species, and usually they reach out to us and say, oh, we're interested in curating some pathways. All the other pathway databases are automatically inferred, and we want to make some specific ones for this or a handful of species. And we say, okay, let's get some starting material. And once there's a handful of pathways, we'll then add the species to Wiki Pathways and get started. A lot of those have ended up with just 4 or 5 pathways, and then the interest has been met or goes somewhere else, and that's just as big as it is. We had a group interested in tuberculosis, and so we curated about 100 pathways with them for tuberculosis, and then it stopped, right? And so they're at different levels, but we don't just automatically infer across all the species. So that's why some of them are much smaller than others.
Can you clone pathways? So, okay.
Yeah, if like across species.
Yeah, like if I wanted to make my own instance or something like that for another species. Though
Yeah.
I think a lot of the value would be having, those merged, but yeah.
Yeah, and so first of all, the simplest clone I'll mention is within a species. So say you want to take apoptosis pathway and make a specific variant of it that occurs in a specific cell type or in a specific disease condition. You can literally just use copy-paste and the tool to copy or just start with the existing pathway, make all your edits, and then upload it as a new one. And so that's one activity that folks do. And then across species, we have a tool that takes a homology mapping to map the gene content, the identifiers from species A to species B. And then we have a little program tool that just runs through and converts all the ones that have one-to-one mappings. The idea is those are the more confident mappings. If there's a one-to-many mapping, we don't make that decision programmatically, and we leave that to the curator to then follow up and either prune out the genes that aren't relevant in that species, or to manually update them to make the clone complete. And then, yeah, you would then upload that to the new species. And so that's something that currently you would just ask us to help you, and we would run that for you. We haven't, if we start getting regular requests all the time, we would make that a public tool, but we haven't had that sort of demand yet. If you wanted to run the tool yourself, we could work with you on that as well. But we're happy to do it at this point because it's a pretty low frequency request.
So, Pankaj here again. I have a question about, let's say, for example, I did a maze pathway curation about a year ago or 2 years ago. And at that time, I was connecting to the ensemble gene identifiers available for that assembly at that point of time. Now what happens is 2 years down the road, things have changed. The identifiers have changed, and how do you manage that? Because, for example, I'm seeing one on the anthocyanin pathway, and those gene product IDs from the list have disappeared. So how do you preserve, although the diagram and everything is preserved, I think the connections to the actual gene IDs are now disappeared somehow.
Yeah, so we probably want to handle that the same way. If we could, if you could direct us to, or someone can identify a mapping between the old and new IDs, I presume Ensemble produces something like that. We could run our tool on that and update it pretty quickly. But we don't have any automated system for doing that at this point.
Yeah, I know, because this is an ongoing black hole kind of situation here, because many times these things happen, and at the most databases are providing mapping to the most previous recent version, but not all the way far down, like 3 or 4 versions back as well, kind of thing. So I think you should think about how you would like to do it. I know human genome annotation is pretty stable and mouse is pretty stable. How do we handle some of the newer genomes that are still in the flux?
Yeah, so this is something where I would ask you, right? So I need to reach out to existing communities that work with those sorts of genomes, because I don't have that experience personally. And how do you do it bioinformatically when you're doing data analysis? How is that? There must be some mapping file. Maybe you have to find all the mapping files and just do them one after another. Mm-hmm. But if that mapping file doesn't exist, there's nothing we can do about it. But if it does exist, we can absolutely update it. But we rely on the community to tell us, hey, this needs to get updated, or we don't have any automated systems for doing updates like that. Mm-hmm. Do you know of services that are provided for species that are changing a lot, that you can ping them once a month and they'll tell you if an ID is changing? I mean, if you know about resources like that, then that can help us design solutions to add automated systems. But I personally don't know enough about that field to.
Yeah, that's where I was a couple of weeks ago. I was at SwissProt, and I think we've just started that conversation.
Yeah,
they also have limited number of hands, and certain ID mapping tools and things are missing.
Yeah,
and there's no mechanism to maintain the mapping files from various different versions that we have. And I can imagine Gramine and Maze GDB and Phytosome may have the Maze genome, but the primary gene IDs all look completely different.
I'm sure we both could go on and on about the harrowing experience with identifiers and ID mapping. I mean, you're describing the case just within an ID system, and then once you want to cross between ID systems, those are changing all the time as well. It's a constant frustration in bioinformatics, and so that's not something we're trying to solve. But if there are solutions out there, we'd be happy to plug them in.
Mm-hmm.
I remember Google was looking at taking over identifiers maybe 8 years ago, or maybe not that long, maybe 6 years ago, and I was hoping some central behemoth would take them over, because then at least we wouldn't have this ongoing issue of trying to convert between them and keep them up to date with groups that don't mind changing their standards constantly.
Right.
Alex, this is Rex Nelson again. I've got a question on how does Wiki Pathway fit into the ecosystem vis-a-vis plant reactome and whatnot? Are you guys seeing sharing pathways? Because if we have a user that comes to Wiki Pathways and takes the time to set up a pathway and show that it's slightly different than maize or Arabidopsis, how does it get around?
Yeah, so we're doing an experiment. We have a great relationship with Reactome that dates way back. And so we're doing an experiment right now with human content. We have all the human Reactome pathways in Wiki Pathways, and we worked with them together and co-published on a converter that goes back and forth. And so we're automatically doing that one. And then if there's edits made to Wiki Pathways that they want to choose, because they have a central curation process to incorporate back into Reactome, they do that. And so it's a nice sharing model there. So we're still testing that out with just one species. We're not doing that yet with other species, but we have the converter. And so if someone wanted to take it upon themselves to convert back and forth, it's possible. But we don't have them integrated back and forth for other species yet.
So Rex, that's where we are working with Alex's group, and there was a group in the Netherlands as well. So we tested it with some of the plant reactome content for reference pathways and I think for maize. But the whole idea was, I think the tool worked beautifully, moving from reactome model into Wiki Pathways. Because the layout and everything was preserved, and I believe it was some kind of and then once the community does some changes on the Wiki Pathway side, we should be able to take it. So I think the channel works backwards as well, but it's the layout issue that we need to address from the Reactome's side. How some of the things, because some of the entity mappings have to be done and the data model mappings have to be done at that point of time. But I think that's certainly a vision that I'm having, at least from the plant reactome side. So for example, we are projecting to 10, 15, or as I said, 74 pathways, 74 species. So if there is glycine max, we can actually take the whole glycine max stack from plant reactome and bring it into Wiki Pathways, and then the soybean curators can actually use and edit those networks that were imported based on homology, and then we should be able to take it back as well. So I think round trip things have to mature a little bit, I believe, on that side.
Yeah.
Okay, just so long as it won't be held hostage in Wiki Pathways.
That's true, and another thing that I've always seen is that when I curate sometimes, for example, and I'm reading and synthesizing all the information before putting it into the plant reactome, because that curated tool is a little bit more complex in plant reactome versus Wiki Pathway. So I curate my thoughts and collect all the data first and put everything in the Wiki Pathways. I know how I'm going to curate. I know all the entities there, and then it's much easier to just cut and paste kind of things to do it in the reactome. So that I've used this strategy as well. So at some point when we release in plant reactome, then those two pathways, those two sources have the consistent form. And I can imagine, let's say, sometime back, I curated the OGS in oxygen transport pathway, and immediately next day somebody came in and said, hey, this is not working, the gene assignments are wrong. And that was based on the publication, but I quickly edited the wiki pathways and then simultaneously edited it in the Reactome as well.
All right, well, I believe we need to wrap this up.
Yeah, I've got folks knocking on my door. I have to go, sorry.
No problem. Thanks again, Alex. I really appreciate it.
Yeah, happy to follow up by email. Hopefully you can share the email if folks don't have it already. Happy
Exactly,
to follow up. And thanks again for having me.
Yeah, thanks, Alex.
Okay.
Thank you, Alex.
Bye.
Bye.
And Pankaj, we're losing people here. Do you still want to talk about your thing, or do you want to wait till next time?
I'll just let people know, and then maybe I'll follow up with an email.
Okay.
Is that I'm organizing the International Conference for Biological Ontologies, August 7 to 10. But before, we are working with Melissa Handel's group to form a consortium or a group on phenotype ontologies and how to curate them, how to build those ontologies and things. And so that's August 6th. And then 2 days after is a machine learning workshop by IBM. If people are interested, you can come in. And while this workshop is happening, or conference is happening, Sushma is organizing a pathway curation jamboree. So it will be a mix of wiki pathways and plant reactome. So I would encourage everybody to come in and join us. There will be, in addition to these, there will be speakers. Obviously, Carolyn is talking, and the folks from PNNL are talking, and then Parag Chitnis, the NIFA deputy director, he is a keynote speaker at the conference. So we would love to see a lot of plenty folks coming in as well.
Great. You know, if you send the